After more than 25 years since acquired immunodeficiency syndrome, AIDS, was first recognized, the human immunodeficiency virus, HIV, continues to present an ongoing challenge for pharmaceutical industry, academic, and government researchers as they attempt to find effective treatments for this devastating infection.
To date, nearly 30 drugs have been released, at least in the U.S. marketplace, and these drugs are used either singly or in combinations. But the treatment of HIV continues to morph because the virus learns how to be resistant.
And despite of an enhanced potency of the more recently introduced treatments, viral resistance continues to be the major challenge for managing patients with HIV.
Taken all together, this brings up an important imperative that new classes of anti-infectives, antivirals, must be discovered to continue to treat these patients.
There is, however, an unexploited target for antiretroviral drugs: the disruption of the use of human transfer RNA, commonly known as tRNA, by the HIV virus during replication. tRNA is essential for the replication and survival of HIV, but until recently, several barriers had prevented exploration of tRNA as a drug target.
The HIV virus actually uses tRNA lysine, and actually tRNA lysine 3. It's very specific on which tRNA it uses, and it uses this to form this RT complex, which is critical for the reverse transcriptase enzyme to actually function. The assay that we have put together will actually use a mimic of the anticodon stem loop of the tRNA that's now been modified by the HIV virus, as well as a piece of viral RNA, and those two are annealed in the assay.
The nucleotide modifications are not only important for selectivity and specificity, but have been shown to increase the binding affinity of this RNA-RNA interaction by several orders of magnitude.
What we are looking for are compounds that just inherently come in and disrupt that annealing process and break them apart. And that is what our fluorescent assay actually demonstrates. And we have now geared this up into a high throughput screening format.
The assay process involves the introduction of probe labeled with a Perkin-Elmer alpha screen fluorescent tag to a solution containing the viral RNA and the respective test compound. If an inhibitor is present, the tRNA will not be able to bind to the viral RNA, and no fluorescence will be detected. The ability to synthesize exact copies of the ASL as they occur in nature gives researchers the ability to methodically search for inhibitors of tRNA. This is particularly effective when screening large libraries of compounds.
In order to find the optimal high-throughput screening assay, we worked with Southern Research of Birmingham, Alabama, to help us understand how best to maximize this assay. Southern Research has done now 3 screenings involving over 120,000 compounds, and in each of the 3 screenings we have found a bioactive hit and have stopped viral reproduction with 51 different compounds. So these have come in each screening that we've done, we've found new bioactive hits.
These chemistries belong to unique families, and because of their unique mechanism of action, constitute a new class of HIV antivirals. Trana Discovery has filed for IP on these families, and all are available for licensing.
Trana Discovery is in a position to license the HIV assay, the class, or lead compounds to qualified pharmaceutical companies for further development. The assay itself can be used to search company-owned compound libraries, confirm the mechanism of action for black hole leads, or screen other commercially available compound libraries.
HIV infections remain a significant cause of chronic and often debilitating disease for patients living with HIV/AIDS, and there is no effective prospect for cure or a vaccine on the horizon. We should emphasize again that therapies that target novel classes, novel approaches, such as the tRNA inhibition, are critical to the ongoing answer to the challenge of this global epidemic.
Through research of the inhibition of tRNA, Trana Discovery opens the way for the discovery and development of new anti-infectives for the treatment of HIV. To learn more about the HIV assay and our other discoveries, visit tranadiscovery.com.